In this lesson, we'll continue our discussion of muscle tissue by taking a look at how a muscle fiber generates tension. And it's important for you to understand in this entire lesson that we're just talking about one muscle fiber, one cell. We're not looking at a whole muscle. We'll do that in a separate lesson. So, depending on what we do, the tension produced by an individual muscle fiber can vary. We're not always going to generate the same amount of tension. And we also know from our previous lessons, the amount of tension that's produced depends on the number of power strokes that are performed at the cross bridges. If you remember, the power stroke is where the myosin head will swivel, pulling the actin towards the center of the sarcomere. All right? And then once the sarcomere shortens, that produces tension. So just to kind of review, the number of contracting sarcomeres in a muscle fiber is fixed, meaning that when a muscle fiber is stimulated, all of the sarcomeres are going to contract, or none of the sarcomeres are going to contract. It's not that one or two contract while the other one's relaxed, right? So all the sarcomeres within one muscle fiber, they're either contracting or they're not contracting, and that's my statement here. So a muscle fiber is either producing tension or it's not, okay? But we can vary the amount of tension that a muscle fiber produces. That's the focus of this lesson. How is that done? There's two things. The resting length of the sarcomere at the time of stimulation is going to be the first thing that we're going to look at. Then we're going to take a look at the frequency of stimulation. If you actually just want to add to your notes, by the nervous system, right? Frequency of stimulation by the nervous system is going to be the second factor that can vary how much tension is produced in the muscle fiber. Again, I want to keep repeating this. We're talking about one muscle fiber at this point. So what we're looking at first is going to be the length tension relationship. Let me read this first sentence and I'm going to take you to a very important diagram. So tension produced by a single muscle fiber relates to the length of the sarcomere, right? The amount of tension produced depends on the number of power strokes performed by the cross bridges, as I said before. But what we're going to look at now is the amount of overlap between the thick and the thin filaments. Let me show you what I mean. I'm going to go skip the slide for a moment and I'm going to take you to a diagram over here. What we're seeing over here, guys, at the bottom is the length of the sarcomere, and this is in micrometers. You don't have to memorize these numbers. It's just more of a concept thing that I want you to get. When the length of the sarcomere is such that it's in an optimal resting length, like I have written down over here, you'll notice the amount of tension, that's this axis right here going up, the amount of tension is going to be maximal. So if you notice, this red line here corresponds to 100. So what we're saying is this: when the length of the sarcomere, remember here's the Z line, here's the other Z line. When this length is a is an optimal resting length, and we stimulate the muscle fiber to contract, we're going to get maximal tension. Let's take a look over here for a second. Let's move to the right. Let's just say this sarcomere, here's the Z line, here's the Z line. Would you agree the sarcomere is stretched, right? Isn't it at a bigger distance, right? It's a wider than this one here that's optimal, right? The distance between this Z line and this Z line here is less than this Z line to this Z line. So that tells me this sarcomere is actually in a stretched position. How can you also tell that? Look at the zone of overlap, right? Only a little bit of actin is overlapping the myosin. Look at the sarcomere at optimal length, right? We have much more overlap. So that means when we have contraction now, right, we stimulate this sarcomere, we have more myosin grabbing the actin and we're going to get more tension. Here, if we stimulate this muscle fiber, this sarcomere only has a little bit of overlap, so now it's not going to be able to generate tension. So as the length of the sarcomere, when it's stimulated, increases, notice the amount of tension we generate is going to go down until, look over here, right? This sarcomere is completely stretched, right? So it's at a big length, right? It's got a great length. But look at the zone of overlap. It's virtually zero. So now when this muscle fiber is stimulated, the myosin heads have nothing to grab onto, and now we're going to be going down towards zero. Okay? So again, what you want to look at is the two things to me are is the distance between the Z lines, right? There's an optimal distance here. That's going to give us 100% tension. If we start to increase the distance of the Z lines, the zone of overlap is also going to decrease. So if you look over here, there's a good amount of zone of overlap compared to here, right? Not as much. So when we stimulate this sarcomere here, we're not going to generate as much tension, and you'll notice the graph goes down. As the sarcomere length gets greater, the amount of tension we generate is decreased. Let's go to the other side. Okay, so again, here we use this always as your reference. Here's the optimal length, but this time, watch this. The sarcomere is going to start in a shortened position, means that this is already somewhat in a contracted state. Look at the Z line on the right to the Z line on the to the left. Right? This sarcomere is shorter than the optimal. Right? So that means we have a greater zone of overlap. Now when you stimulate this here, right, there's going to be less sliding filament taking place, and notice we're going to generate less tension because it's already in a shortened position, and you'll see the curve go down on this side, meaning that if we stimulate a sarcomere that's already shortened, it's going to generate less tension, and the more shortened it is upon stimulation, the less and less tension we're going to get. Look at this one. This is maximally shortened. This is already at full shortened length. Look at the actin. It's already crossed over the M line. So if you stimulate this sarcomere here while it's already in a maximal shortened position, you're going to generate no more tension. Okay? So the concept is this. We generate the most tension when the sarcomere length is at the optimal distance, let's say, and we have good amount of zone of overlap. If the sarcomere length is increased with less zone of overlap, the tension goes down. Or if the sarcomere length is decreased and we have too much zone of overlap, we are also going to get less tension. All right, so it's an interesting picture to look at. Once you sit back and dissect it, you'll see it actually makes sense. So let's summarize what we're talking about. All right, so optimal resting length, and again, you don't have to know the number of micrometers. I'm not going to ask you that. Just kind of know that there's an optimal resting length. Here we're going to have a good zone of overlap, right? The zone of overlap is going to be large, and we're going to produce maximum tension. But if we increase the sarcomere length, you'll notice, again, if you go back to the picture, the zone of overlap is decreased, right? We increased the sarcomere length here. We stretched it, it's kind of like stretching the muscle fiber, and we pulled the actin away from the myosin so that it's not as much. The tension is also going to decrease, right? So the more we increase the length, the more the tension is going to go down. And just the opposite, if we decrease the sarcomere length, right, we start already with a shortened sarcomere and now stimulate it, we're also going to get less tension, right? The thin filaments can actually extend across the center of the sarcomere at the M line, and the tension is going to decrease. That's this side over here. And so again, we have a sarcomere here. It's much shorter than the optimal. Right? We have a lot of zone of overlap, but now when we stimulate it to contract, right, we're not going to get more zone of overlap, so we're not going to generate as much tension, and the tension will go down. Okay, so this is one major factor that determines how much tension we can generate in one muscle fiber. Right? It is called the length tension relationship. Okay, so the second concept I wanted to talk to you about is known as the frequency of stimulation. And again, you may want to add here to the notes by the nervous system. I kind of assume people know that, but you may not. When we talk about frequency of stimulation, right, whenever we're stimulating something, we're talking about by the nervous system. So what we have with a muscle fiber, if we stimulate one muscle fiber once, like one nerve impulse to a muscle fiber, we get something known as a twitch contraction. Right? So it's going to be a contraction that generates tension, and the single neural stimulation produces a single contraction and then relaxation. Now, in real life, this is not enough to generate enough tension in the muscle fibers or a whole muscle to do the work that we need to do. But I want to teach you this basic premise so that we can build on it. And you can see here, depending on the muscle fiber in the body, it can last anywhere from 7 to 100 milliseconds. And there's different factors about that, but one of the important factors is the location of the muscle. For instance, we see that eye muscles and hand muscles, they have a faster twitch contraction. They're going to be closer to the 7 to 10, say, milliseconds. So these muscle fibers have a much quicker twitch contraction, and they can move much faster, opposed to like a lower leg muscle like the gastrocnemius or the soleus muscle. We're going to see that they have a much longer twitch contraction, again, because they have different types of functions. These are more postural muscles, believe it or not. So we're going to take a look at the parts of the twitch contraction. When we have a sustained muscle contraction in our body, right, say so we have a muscle fiber that's going to have a long sustained contraction, one single nerve stimulus is not enough to generate a sustained muscle contraction. We actually need repeated nerve impulses going to the muscle, and that's what I'm going to show you, or actually going to the muscle fiber. We can graph this out on a special graph known as a myogram that shows us the tension that develops in the muscle fiber. Again, I'm going to remind you, we're just talking about one single muscle cell. We're not talking about a whole muscle like the biceps. We're just talking about one cell, let's say, within the bicep. Okay? So I'm going to jump over this for a minute, and I'm going to show you something. What we have here, this is an actual myogram of a twitch contraction, that red line. And what we see down over here is the time in milliseconds. And then over here, the amount of force that we generate, the tension. So this green arrow kind of represents the point that one muscle fiber was stimulated. Let's say we were in the laboratory and we hung up on a hook one muscle fiber, and we took a little electrical wand, right, and stimulated that muscle fiber. This is the twitch contraction we would see. The green line represents the point that we touch the muscle fiber. All right, so this is going to be like ground zero, point zero. So notice when we stimulate the muscle fiber, there's a period of maybe 3 to 4 milliseconds where there's no tension being generated, right? It's kind of like just a flat line right here. This period is known as the latent period. Right? When I was in school, we learned it as the lag phase, LAG, and it's just a period of time where between the stimulation and when we start to generate tension. So what's happening there? In the latent period, when we stimulated that muscle fiber, the action potential, right, which is the nerve impulse, was traveling along the sarcolemma. So that takes a little bit of time. Then the sarcoplasmic reticulum has to release the calcium, and that calcium floods the sarcomere. So no tension is produced. So it's important to note during the latent period, right, activities are happening within the muscle fiber, but there's no tension being generated. Then at some point here, it looks like 3 to 4 milliseconds, the curve starts to go up and the muscle fiber starts to generate tension. This is the second phase known as the contraction phase. So at that point, the calcium is binding to the troponin. Remember, the troponin changes its shape. It pulls the tropomyosin out of the way, exposing the active sites on actin, and then the myosin heads jump up and they form the cross bridge, and then they swivel, and we get the sliding filament theory, and that's what generates the tension, and the tension begins to peak. So I circled it over here. The contraction phase, right, started after the lag phase, and it goes about 10 15 seconds. Again, it's going to be variable, right? So this period of time right here, this is the contraction phase, right? That's this curve going up all the way to the peak, right? So everything under here is going to be contraction time. All right, so we're getting the sliding filament theory happen. The final phase is going to be the downward slope here, this red line going down. This we call the relaxation phase. At this point, there wasn't any more nerve stimulations. We only had the one nerve stimulation. So the calcium is going to be released from the troponin. The calcium gets pumped back into the sarcoplasmic reticulum. The cross bridges detach. The active sites become covered again by the tropomyosin, and the tension goes down. And that's the third phase I want you to know, known as the relaxation phase, and that's this swing down over here. And again, the reason this happened, the up and then the down, is because we only stimulated the muscle fiber once. We gave it one stimulation, so we got one contraction and a relaxation. So here, this is just kind of showing you, you don't have to know these times, just to kind of give you an idea how like an eye muscle, notice the twitch contraction is relatively quick, right? It's, maybe 10 between the lag and the when it stops, end of relaxation, maybe 15 milliseconds, right? Our eye muscles move very, very rapidly, where the gastrocnemius in the calf, that big muscle in the back of your calf, has a longer one. This one goes about, say, 40. And then the soleus muscle, right, goes like about 100, over 100 milliseconds. So each muscle has a different characteristic twitch contraction. You don't have to memorize this. I just want to show you that not all twitch contractions in each muscle is going to be the same. Okay. So, like I said before, in the body, when our muscle fibers are stimulated, they're not just stimulated one time by the nervous system and we get a twitch contraction where that's enough to do work. It turns out we have to repetitively stimulate the muscle fiber to get a good amount of tension generated. So I want to kind of lead to you to what the final type of contraction is going to be, called uh, a tetanic contraction, a tetanus contraction. That's where I'm going to lead you to. But before I can teach you that, I have to show you some basics. There's a concept known as trepa. This is a German word for a stair step, like a staircase, increase in tension. So I'm going to skip the notes for a minute and show you here. Watch this. Let's see if you can recognize. So here we're showing you the green arrow represents the stimulation of the nervous system. Again, this is something we would actually see more in the lab. Skeletal muscles don't even behave like this in the body because this is kind of like more of a physiological concept. So let's just say we hung a muscle fiber up in the lab and we stimulate it right here. What we're going to get is the contraction phase and then the relaxation phase. Notice that's a twitch contraction. That was this, right? Contraction phase, relaxation phase. Then, once the muscle fiber relaxed, guess what we do? We hit it again with another electrical stimulation, and we get another twitch, contraction phase, relaxation phase. But notice the peak of this second curve is higher than the first. So that means when we stimulated it a second time, after it relaxed, it was able to generate more tension. Right? So this went through contraction, relaxation. Once the muscle fiber relaxed, we stimulate it again, and now we get a third curve, and it gets a little bit higher. And we continue to do that. We stimulate the muscle fiber after each relaxation. And we notice, see this kind of like a staircase stepwise increase right here. That's where they get the term trepa from. Like just like that. And then at some point it plateaus out. We get maximal contraction. So notice it's the maximum contraction for trepa, right? We don't get maximal contraction of a whole muscle fiber. We're getting maybe a quarter of what it should be. Right? So this is what we call trepa. Right? It's a stair step increase in tension. It's caused by repeated stimulations, highlight right here, immediately after relaxation phase. And you'll notice the frequency of stimulation is probably less than 50 stimulations per second. So this produces a series of contractions with increasing tension, but at some point it levels out. We never get to 100% tension, right? We only get maybe a quarter of the way up, all the way. So we don't really see this in skeletal muscles. Our skeletal muscles don't behave like this. This is something we would definitely see in a skeletal muscle in the laboratory. But cardiac muscle in our heart, they may do trepa. Right? And that's a little bit beyond the scope of our course. The concept I want you to know, though, is that as we increase the frequency of stimulation, and again, the stimulation is happening right after the twitch contraction relaxation phase, we're going to get a slow increase, and then at some point it's going to level out. But again, maybe 25% of like maximal. We never get 100% up here. So this concept is known as trepa. So now what they did in the laboratory, they increased the frequency, right? We were going to make these electrical stimulations even faster. Watch what happens. When they did that, they found that they could actually marry two waves together in a concept known as wave summation. Again, I'm going to skip the note and I'm going to take you to a picture. So watch what happens here. Here's our first stimulation. We get the upward curve. That's the contraction phase. And then the curve starts to go down. That's the relaxation phase. But watch this. They don't let the relaxation go all the way to the bottom. While the curve is going down, while the relaxation phase is happening, they stimulated the muscle fiber again. But notice now we got a much higher curve than for the next curve, the second one. So we actually summated, we actually added the waves together to get a much stronger curve or a much higher amount of tension. Right here we're already up to say a quarter of 100%, right? If this is 100% up here, and we're about at a quarter. So now this second wave is occurring as it's going through relaxation. We hit it again, and now the third curve is even higher. And then the fourth one is even higher. So this concept is known as wave summation. This occurs when successive stimuli arrive before relaxation has been completed, right? We're hitting the muscle fiber while it's in relaxation. Okay, so this is wave summation. I'll go back to the verbiage for a minute. This is an increasing tension due to the summation of the twitches. Right? It's caused by repeated stimulations, highlight right here, before the end of the relaxation phase. Right? In order to do this, we have to stimulate it more than 50 times in a second, that muscle fiber. Right? Remember with the trepa, it was less than 50 times in a second. So it turns out there are two types of wave summation. The first type is called incomplete tetanus, and the second one is complete tetanus. Complete tetanus is the way our muscle fibers actually behave when we produce our movements to generate full tension. So let me show you the difference. Let's take a look at incomplete tetanus, right? Notice, guys, we're increasing the frequency of stimulation, right? We're going to make the stimulation quicker and quicker. All right, so it gets so fast that we marry the waves together. We get wave summation. But again, at some point, it starts to level out. Maybe at 75% of maximal tension, right? So incomplete tetanus occurs if the stimulus frequency increases further. Right? We're going, a little bit higher than 50 times per second. The tension production rises to a peak, right? It's going to plateau off right about over here. Okay, and you still have some periods of relaxation, right? So you're still seeing some relaxation. This is known as incomplete tetanus. But now, if you really crank up the frequency of stimulation, notice eventually you don't see any more relaxation. The frequency is so quick, right? We're stimulating this muscle fiber so fast that we end up getting a plateau here at 100% maximal tension, without any more periods of relaxation. Notice what's missing, right? We don't see these downward slopes anymore. They eventually disappear, and we get a nice sustained contraction. This is known as complete tetanus. Right? During complete tetanus, the stimulus frequency is so high that relaxation is eliminated, and tension now is going to be at a maximum level. Okay, so just to go back to the notes, incomplete tetanus, the muscle produces near maximum tension, right? We get close to about maybe 75% of maximum, and we're going to see that there's rapid cycles of contraction and relaxation. We still get some, again, contraction and relaxation. Compared to complete tetanus, the stimulation is increased so much that eventually relaxation is gone, it's eliminated, and now we get a more continuous contraction. Okay, and this is the way our muscles contract, on a regular basis to produce movement. All right, so that's the way our nervous system is stimulating the muscle fiber. So that concludes this lesson on how we generate tension. And again, it's important for you to remember how we generate tension in a muscle fiber, a muscle cell, not a whole muscle. We'll do that in the next lesson. And what we looked at was the length tension relationship, which is the length of the sarcomere when it is stimulated. And then we looked at the frequency of stimulation by the nervous system to ultimately give us complete tetanus.